of you who haven't met me before, I'm one of the clinicians working on Alzheimer's disease research, particularly familial Alzheimer's disease research here at the Dementia Research Centre. So today I'm going to talk about neuroimaging in the diagnosis and understanding of Alzheimer's disease. So please feel free to interrupt me as we go along. I'll try and avoid too much jargon, but we do have a habit of occasionally slipping into that, so I'm happy to take questions either at the end or as we go along. I don't want you to be sitting there not knowing what I'm talking about. Okay, so I'm going to begin by discussing the changing role of imaging in the clinical diagnosis of Alzheimer's disease. I'll talk about the different types of brain scan we have available to us and how these allow us to investigate the disease from a range of different perspectives, from structural to functional to molecular. I'm then going to focus on two ways in which imaging is being used to increase our understanding of familial AD in research. Firstly, through exploration of the clinical phenotype of familial AD, and by phenotype, I mean the way it presents in an individual, the types of symptoms, the types of neurological problems that an individual might experience. And then I'm going to talk about the use of imaging to investigate the long preclinical phase that we now recognise exists before symptoms start, and how in doing so it's facilitating trials of new treatments, including pre-symptomatic treatment trials. So starting with the clinical diagnosis, so guidelines from both Europe and America state that all patients suspected of having dementia should have brain imaging with a CT or MRI at least once. Now, historically, this was in order to exclude an alternative cause of their dementia, so a tumour, as you can see in this case here, hydrocephalus, where there's a buildup of fluid in the brain, or a bleed like a subdural hematoma. So in our clinics, we rarely see these pathologies, but they're so important not to miss because they're potentially treatable with surgery. But increasingly, it's recognised that imaging offers not just exclusion of alternative pathologies, but positive information to support a clinical diagnosis of Alzheimer's disease. So the European guidelines go on to state that to increase the specificity of diagnosis, MRI should be used above CT. And on MRI, we can assess hippocampal atrophy. So this means, well, atrophy means loss of brain cells, and the hippocampus is a particular area of the brain that's very important for memory. So assessment of hippocampal atrophy is something that clinicians look at on a scan, and if it's present, it can provide a lot of support for a clinical diagnosis. And the guidelines also state that when the diagnosis is unclear, additional types of scanning, two types of functional scan called FDG PET and SPECT, which I'll talk about a bit later, they may be useful. And again, when the diagnosis is unclear or to document disease progression, repeating the scan, the MRI scan after a period of time may be helpful. So what do we look for on MRI? Well, first of all, we look for any tumours or bleeding, these rare structural causes of cognitive impairment that could be treatable with surgery. We then look for any vascular pathology, so pathology of the blood vessels. Are there any infarcts suggesting a previous stroke? Are there changes to the white matter? I'll talk about this more later, and it can be caused by lots of things, but most commonly white matter changes are seen when someone has vascular risk factors, so things like high blood pressure, high cholesterol or smoking, and that can all cause cognitive impairment. And we look for something called microbleeds, so as the name suggests, these are very small areas of bleeding. They don't cause symptoms, but if they're present, then they can provide important clues to the underlying diagnosis. So if you can see on this scan here, sorry it's not projecting very well because we don't have any blinds, but I don't know if you can see, there is some tiny black dots towards the middle of the brain. Can people see them? So they're microbleeds. And in this scan, they're very deep in the brain, and a deep pattern of microbleeds is most commonly seen in vascular disease, so most typically due to high blood pressure. Whereas in this scan, again, they don't come out very well, but there's lots of little black dots around the edge of the brain, so they're much more peripheral. And that distribution is more commonly seen in something called Cerebral Amyloid Angiopathy. It's a bit of a mouthful, so we usually say CAA. And what that describes is amyloid, the abnormal protein that is deposited in Alzheimer's disease, but deposited in the blood vessels. Now, about 90% of patients with Alzheimer's disease have some degree of CAA, if we were to look in their brains at post-mortem, but the degree really varies between individuals. So we then move on to look for atrophy, this brain cell loss. Is the generalised atrophy. Now, unfortunately with age we all lose brain cells, so the amount of brain cell loss you see depends on the age of the patient. In a 98-year-old, you'd expect to see quite a lot of volume loss. Whereas if you saw that same degree of shrinkage in someone who was, say, 40, then you'd be much more concerned that there was a degenerative disease present. And then we look for brain cell loss in particular regions. So, as I said, we look at the hippocampus, this important memory area, and we look at the posterior cerebral hemisphere. So this is areas at the back of the brain. In Alzheimer's disease, we tend to see more atrophy of the hippocampus in the back of the brain, whereas in frontotemporal dementia, the other disease group that we shared the meeting with this morning, you'd expect to see more brain cell loss in the front of the brain, so the frontal and temporal lobes. So hippocampal atrophy is the most well-established imaging marker of Alzheimer's disease. So this is the hippocampus, this area here. Hippocampus means seahorse in Latin, and so I think you can maybe convince yourself it looks a little bit like a seahorse lying on its side. And these are just some of the many papers that have described an association with hippocampal atrophy and Alzheimer's disease. And even in mild Alzheimer's disease, the hippocampus is up to 20% smaller than in control. So it's not surprising that people have memory problems when you see how this structure is affected. Sometimes it's not that clear though at the initial scan, and so in those cases, for example, if someone has memory symptoms and there's a lot of anxiety and depression, and this is very common if someone's got a family history of the disease, when we first see them it can be unclear whether it represents the beginning of the disease or actually if they're having difficulty with their memory because they're anxious and depressed, because those factors really affect how we retain memories. And so sometimes in those circumstances, it's useful to repeat the scan after an interval. So you can see in this scan here, so the yellow area shows the hippocampus, so it's enlarged here. So it's possibly a little small at baseline. You can see quite a lot of black space around it. But then when the scan is repeated at 18 months, it's actually quite a lot smaller. You can see a lot more space around it. Then when it's repeated again at 36 months, it's even smaller. So sometimes it's only over time that it becomes clear what is evolving. Again, sometimes when the diagnosis is uncertain, we move to doing extra scans. So this is functional imaging with something called FDG PET. So this is a marker that's injected into the vein, and it looks at glucose metabolism in the brain. So the areas of red show areas of normal glucose metabolism, and you can see in this control, so a healthy individual who's participating in research, they've got very widespread glucose metabolism. But in Alzheimer's disease, on the second row, you can see it's decreased in the back of the brain. This is the brain looking from top down, and this is the brain that's viewed from the side. That's the front, that's the back. And in FTD, frontotemporal dementia, we see decreased metabolism at the front of the brain. Okay, so they're the imaging modalities, the types of scan that we use in clinical practice. But in research, we have a whole range of additional scanning techniques available to us that are not yet well enough understood to go into clinical practice, but can provide insights into the pathology from a range of different perspectives. So the pathology of Alzheimer's disease is deposition of amyloid, this abnormal sticky protein, either in plaques in the tissue or in blood vessels, and the deposition of a protein called tau in neurofibrillary tangles, and this structure is indicated by the arrow. So we can look at the effects on the structure of the brain with MRI. We can look at effects on the function with SPECT, FDG PET, and particular types of MRI that look at perfusion or function. So some of these functional MRIs where individuals lie in the scanner and we measure brain activity either at rest or when someone's doing a particular task like a memory test in the scanner. And there are advanced types of MRI scanning, so something called DWI, diffusion weighted imaging, and that allows us to look at the microstructure of the brain by looking at the diffusion of water in brain tissue. And then an even more complicated version of that technique is something called diffusion tensor imaging, DTI. And that takes that information about diffusion of water and allows us to build up a picture of the three-dimensional tracts that connect different brain areas with each other and to take information from the brain to the spinal cord. We get these beautiful images that provide information about the connectivity of the brain. We can use specific scanning sequences to look at certain pathological effects. So these are those microbleeds I was mentioning on a certain type of scan called T2 star. And then there is molecular imaging, and that's where something is injected into the vein that binds to a particular molecule. So we have traces that bind to certain neurotransmitters like dopamine, and that bind to amyloid, so we can actually look at amyloid deposition, at tau deposition, and even at markers of inflammation that bind to specific markers on, for example, microglial cells, which are important cells in our immune system. So the development of amyloid imaging over 10 years ago now was a major breakthrough in Alzheimer's disease research, because for the first time researchers could see in life the areas where this abnormal protein is deposited. And these are scans from a patient with Alzheimer's disease, and the red show areas of amyloid deposition. You can see it's really quite widespread, whereas this healthy control has no amyloid deposition. And the most widely used tracer for these scans, or ligand, is something called PIP, which stands for Pittsburgh Compound B. It was developed in Pittsburgh. Now, use of this tracer was always going to be quite limited because it uses radio-labelled carbon, so it has to be made in a big machine called a cyclotron, and it has to be made at the site of the scan because it breaks down relatively quickly. So for us, currently the nearest site for this is Cambridge, and any of you who participated in our amyloid imaging studies will know that it's quite a high-maintenance type of scan, because if anything goes wrong with the cyclotron, when the compound's being produced, or with the scanner, then that whole scan has to be cancelled. Each tracer is made on the day for that particular scan. So that was never going to be a type of scanning that could become widespread in clinical use. But more recently, a number of newer compounds that use a different compound labelled with fluorine have been developed. They don't need to be produced in a cyclotron, and they have a much longer half-life. So we're beginning to see that their use is much more widespread. And in fact, 3 of these such tracers have now been approved for clinical use in the States. So even more recently, just actually in the last couple of years, new tracers have been developed that actually bind to tau, the other abnormal protein that is deposited in the disease. And so we're now able to start looking at how the deposition of these 2 proteins relates and differs from each other. So here you can see in a patient with Alzheimer's disease, so that's their MRI, that's their tau scan in the middle with this compound PBQ3, and that's their amyloid scan with PIP. So the areas of hippocampal atrophy are accompanied by quite marked deposition of tau, whereas amyloid is very widespread, but not actually so much in those areas. So over the coming years, I think this is going to help us to really understand a lot more about the disease process. And our very own Phil Weston has recently successfully applied for funding to pilot the use of tau imaging in familial Alzheimer's disease. So this is a really important development. So the first application of imaging that I'm going to talk about is how it can be used to investigate variability in the clinical presentation of familial AD. And I want to talk about this for 2 reasons, partly because it's something I'm very interested in, but also because I think it's a really good example of how the questions that you ask us, the things that often we can't answer, and the subjects that we discuss at these meetings can sometimes directly inform the things that we go on to look at in research. So over the years, a lot of you have asked me questions on this theme. Why do these mutations that cause familial AD, why is it that they can affect different people in such different ways? So we see variability in the age at which people develop symptoms. We see variability in disease duration. So some people, unfortunately, when they develop symptoms, they can progress quite rapidly, whereas other people can have very mild memory problems that remain relatively stable for many years. And so when people ask us the inevitable question, how long will this last, we really can't answer that at the moment, because we see just how variable the disease can be in different people, and we don't yet understand why that is. But if we could understand it, for example, if we could understand what makes someone able to have a much more slowly progressive disease, then perhaps that could provide clues for how we could develop better treatments. So we also see variability in the initial symptoms. Most people have initial problems with memory, but sometimes language can be affected, or behaviour first. And some people have additional neurological features. Some people develop seizures, not everyone does. And some people remain physically very active throughout the disease, whereas others have a lot of difficulty with walking due to leg stiffness from really quite early on. And we see variability between the different genes that cause Alzheimer's disease, the 2 commonest being APP and presenilin 1. We see variability between different mutations within these genes. And we even see variability between, and even within, some families with the same mutation. So imaging can sometimes provide insights into possible sources of this variability. So this is a study we did a few years ago. We wanted to see if the patterns of grey matter loss and white matter loss were different between people with mutations in APP or presenilin 1. So in this type of image analysis technique, all the scans of symptomatic patients with APP mutations are compared with controls, and all those with preselin1 mutations are compared with controls. And the red and yellow areas show areas of significant volume loss in the patients compared to healthy controls. And so you can see in the preselin1 group here, there's quite widespread loss of grey matter volume, whereas in the APP group it's less diffuse and it's more focused on these hippocampal areas. It's more obvious in the white matter. There's quite diffuse loss of white matter, whereas in the APP group it's much more severe but focused in the hippocampal area. And that's interesting because most of the patients we see with APP mutations have initial and very prominent memory symptoms. And whilst patients with preselin1 also often do just have memory problems, they can also have additional areas affected early on, so behaviour, language, and sometimes there's things like the difficulty walking I described. So I became very interested, particularly in this white matter change, because it's historically been the grey matter that receives the most attention in Alzheimer's disease research. And a few years ago, a new imaging technique was developed that allows us to look at the microstructure of white matter. It's called Diffusion Tensor imaging, or DTI. So this was a study we published a couple of years ago when we looked at the white matter microstructure in patients with preselin1 mutations, and we found that symptomatic patients have this very widespread loss of white matter integrity, and these different metrics, fractional anisotropy and axial, radial and mean diffusivity, and they basically tell us about the way the white matter is damaged in different ways, so they give us insights into the underlying pathology. But again, this is looking at a group level. So is there anything that we can see on an individual patient scan that tells us whether the white matter is affected? Well, in some cases there are. So there's something called white matter hyperintensities, and I'll show you on this scan here. Can you see these blobs of white? So that's a white matter hyperintensity. You can see there's a few scattered at the front there, they're mainly at the back. So these are a very non-specific imaging feature. We see them commonly in patients as they get older. They're thought to reflect generalised wear and tear of the blood vessels, and they're much more common if people have high blood pressure or smoking or other vascular risk factors like high cholesterol. But I was noticing them in people with familial Alzheimer's disease who were young, often didn't smoke, didn't have high blood pressure, didn't have high cholesterol. So I started thinking this must tell us something about the underlying disease. And so one of the other pathologies that can cause white matter hyperintensities is this CAA, Cerebral Amyloid Angiopathy, when there's amyloid in the blood vessels. And there's some work from the pathology literature that suggests that certain preselin1 mutations located in a particular area of the gene, so after codon 200, have more of this CAA. So preselin1 is a very big gene, and there are nearly 200 different mutations in it. So I'm not sure how well it's projecting, but this is a diagram of the whole preselin1 gene. The red circles show areas where a mutation has been identified. So they're scattered throughout it, and codon 200 is here. So we looked at whether people with mutations after codon 200 had more white matter hyperintensities on MRI than those before, and we found that they did. And for this study, we were able to look at scans collected really over the past 20 years from families who participated in our research. And some individuals had subsequently died, and 10 had made the very generous decision to donate their brains to research after death. So working with Tamara and Lashley, who you'll be hearing from next, we were able to look in those brains and see what actually was the severity of cerebral amyloid angiopathy, the deposition of amyloid in the blood vessels. And we found that it did correlate with the severity of these white matter hyperintensities. So we've just finished this study, and I think it's important because it shows how something that we see on imaging can really provide us with insights into what's going on pathologically. And now that there are so many new treatments being developed, it's possible that some people will respond better to certain types of treatments and other people to others. We just don't know yet. But the more we can understand at this stage about variability between the different mutations, the better position we'll be in to interpret the results of these trials when they start to become available. So we'd like to do a lot more of this kind of research in the future. Research where we start by looking at how people are different in terms of their clinical presentation. We then go on to look at their scans to see does that provide insights into this difference. And then we look in a range of different ways at the pathological and molecular mechanisms that might underlie this difference. So we can look in the blood, we can look in the cerebrospinal fluid. And for people who are do feel willing to make that decision to donate their brain to research, we can look at an awful lot of things at a pathological level. Now, Tamarin will speak more about this later, so just to mention a few things, we can look at the deposition of amyloid in the blood vessels. So over here, this is a blood vessel. The dark brown material is the amyloid protein, and these are some amyloid plaques. And it's you can actually see different types of plaques in different individuals. So some people have these things called cotton wool plaques because they're a bit fluffy. We don't quite know why they develop these, but it's something that we're starting to look into, and it could provide us with important insights. Just as we can look at the white matter on scans, we can also look at it in the brain. So these streaky lines are white matter fibres, so we can actually look at how the pathology is affecting the white matter. And we can look at the role of the immune system by studying cells that are involved in immune and inflammatory function. And we can go on to look at the molecular level by looking at the different types of amyloid that are there. So, like all things, the more you start to learn about something, the more complicated it turns out to be. So amyloid is this sticky protein that's deposited, but it's actually made up of proteins of different lengths. So of 38 amino acids, 40, 42, 43, different lengths of amyloid that then clump together. And we can actually look at the relative amounts of each one. And that may have an effect on the way the disease manifests and the type of pathology that we see. And the other way that we can gain insights is through skin biopsy research. So many of you will remember Selena Ray, who leads this research, and she came and gave a brilliant talk here a few years ago. And she's running the marathon tomorrow, actually. We loved it. So she does this incredible research from just a tiny skin biopsy taken at the upper arm of someone with a mutation, she's able to take those cells, those skin cells, primary fibroblasts, by growing them in a special soup, and turn them back into the pluripotent stem cells that could have become any cell type in the body. And then by growing them in a different type of soup, turn them into neurons, so that the actual brain cells that she can then study. And there are a whole host of things that she can look at in them to see what the mutation is doing. For example, looking at how it affects different amyloid structures. So we can sort of bring together all these different types of research to understand the mechanisms that lead to the disease. And the other incredible thing about these cells is that they offer a model where we could potentially test new treatments before they go into patients. So that's one way in which I think imaging is important. The other way I wanted to talk about was in better understanding preclinical Alzheimer's disease. So this is a hypothetical model, a predicted model of how scientists think Alzheimer's disease markers may evolve over time. So there's now quite a lot of evidence that this suggests that there's a long phase during which pathology accumulates before the onset of symptoms. So that's the preclinical phase. When someone develops symptoms, they're said to enter the mild cognitive impairment phase, and then once those symptoms reach a stage of impacting on their function in everyday life, they're said to have entered the dementia phase. So a lot of different strands of evidence suggest that people can have a lot of amyloid in the brain without it causing any problems. So, for example, 30% of healthy older people who volunteer to take part in research with amyloid scans are found to have positive amyloid scans. And about 30% of healthy elderly people who die are found to have amyloid pathology at post-mortem without any symptoms. So what we don't know is the proportion of these that would eventually go on to develop symptoms. But it's thought that eventually it causes significant damage really to be detectable on functional imaging, then to be detected on markers of neuronal injury in the CSF, and eventually to cause changes in brain structure. So I imagine that seeing these sorts of images is probably quite daunting to some people, the idea that there are changes that we can detect before symptoms develop. But I think there's also a very positive way of looking at it, because if we can detect changes that early, then we have targets that we could treat and ways of seeing if our treatments are working. So the idea is if you could stop the amyloid accumulation at some point along this line, then ideally we could hopefully either prevent or delay or totally prevent the downstream effects. And so that is the aim of research moving forward. And obviously familial Alzheimer's disease and people at risk of familial AD provide a really unique opportunity to learn more about this preclinical phase, and people have been incredibly generous with their time in participating in research. So most of you will have heard of the DIAN, Dominantly Inherited Alzheimer Network study, which is collecting imaging and biomarker information from people who are at risk of familial Alzheimer's disease, who are much younger than the age at which they would be expecting to develop symptoms. And this is some work from DIAN that was being published and suggesting that indeed amyloid imaging changes are detectable about 15 years before the expected age at onset. Then changes in functional imaging are detectable later, and cortical thinning detectable even later. And this is cross-sectional work, so it's implied that these changes are progressive, but it's actually just looking at the baseline scan of individuals who are predicted to be a certain distance as measured by the parent's age at onset. And so we don't know how accurately that does predict things. But the DIAN study also showed that when looking at some brain areas, so subcortical areas, areas deep in the brain, different areas seem to show different vulnerability to pathology. So we see decreased metabolism in the hippocampus, but other areas seem to be able to tolerate a lot of amyloid for a long time without causing any problems. And also, there seems to be a suggestion that very early on, some areas are actually hypermetabolic, so they have increased activity. And we saw a similar thing when we looked at diffusion. We found that some areas, they have altered diffusivity in one way when somebody is symptomatic, but in another way in the pre-symptomatic phase. And this is important because it means that these imaging modalities can sometimes detect changes before degeneration. And these are really the changes we'd like to be able to detect in treatment trials. We want to see, are we stopping pathology before it causes these later degenerative effects? And so we're starting now to see the results of longitudinal studies, and historically the imaging marker that's being studied the most longitudinally, so this is looking at scans of the same person over time, has been this hippocampal atrophy, so volume loss in the hippocampus. This was a study by Nick Fox and Basil Ridder where they looked at rates of hippocampal volume change in mutation carriers and controls. And you can see this is why it's so important to have healthy controls in research, and a lot of these are the controls in blue, a lot of these individuals were siblings who'd had negative tests, because you need to know how much brain cell loss declines over age, because I said in everybody it does decline a certain amount. But you can see that the lines in red which show the mutation carriers, it's declining a lot faster. And when these results are sort of modelled together, this is this sort of rate of decline slope, and we can see changes become detectable between mutation carriers and controls about 3 years prior to diagnosis, and by the point of diagnosis, here the hippocampus is about 20% smaller than in controls. We're able to pick up changes even earlier when we look at it in an individual's own rate of decline. So here, this is instead of looking at differences between the groups, but just looking at one individual's rate and comparing it to another individual's rate. Here we can detect changes up to 5 years prior to expected to the clinical diagnosis. So you can imagine an era of treatment trials where ideally you would know that someone had amyloid pathology because they had a positive scan. You would follow them over a period of time to see what their atrophy rate was, and then at a certain point you would initiate treatment while they're still well. Scanning would then have two roles. It would be important for safety, so you would want to make sure that the treatment wasn't causing any effects, for example causing new microbleeds on their scans, but you would also use scans to look for effects of the treatment. So is there a decrease in the amyloid deposition? Or does this slope of brain cell volume loss slow down so that it approaches the slope that we see in healthy controls? Well, that era has we hope, has arrived. So over the last year, a number of preclinical prevention trials have started, and the people in this room who are some of the first people in the whole world to enter these trials. So the DIAN study is doing a trial of 2 antibodies against amyloid. The Alzheimer's Prevention Initiative is, I need to update that, it's not longer than 2 years. The Alzheimer's Prevention Initiative is looking at a large family from Colombia with a certain presenilin-1 mutation using a different amyloid antibody. And the Alzheimer's Prevention Initiative also has a study looking at healthy older people who are at risk of sporadic Alzheimer's disease because they have 2 copies of the ApoE4 allele, and that's the commonest genetic risk factor. It's not a causative gene, but it's a risk factor for sporadic Alzheimer's disease. And then there's something called the A4 trial in the States, which is studying 1,000 cognitively normal older people who have positive amyloid scans. So all of these trials use different types of amyloid-modifying therapy, and there are a lot more in the planning stage. So it's exciting times in research, and I think there's been a lot of low morale recently because people are always reading the headlines, another drug trial failed in Alzheimer's disease. And I think it's important to remember that while these previous trials in patients with symptomatic Alzheimer's disease failed to show a clinical benefit, they did sometimes show evidence of an improvement on imaging. So these are amyloid scans from a trial of an antibody against amyloid called bapineuzumab. On the bottom here, you can see scans of 2 individuals who received placebo. At baseline, they both have amyloid deposition, these areas in red, and then at follow-up, they've got even more amyloid deposition. Whereas in these 2 treated patients who received bapineuzumab, at baseline, they've got amyloid deposition, but you can see there's quite a marked decrease in the amount of amyloid by their follow-up scan. So I think these are really encouraging results. And so I'd like to end on that note of cautious optimism. So in conclusion, we've discussed how imaging offers positive contributions to the diagnosis of Alzheimer's disease. Different types of brain scan provide insights into a whole range of different aspects of pathology. Imaging may be used to enhance our understanding of variability in the clinical presentation of the disease, and it reveals the preclinical periods when changes progress and evolve, and this itself opens a window for clinical trials and the possibility of disease slowing to delay symptom onset. So I'd just like to end by thanking my colleagues, funders, and most importantly, everyone who participates in research, and thank you all for coming along today. I'm happy to take any questions or to be contacted at any point with any questions.